Okay, hello everybody. Welcome to lecture 4 of genetics. And we're going to start out this lecture with talking about how we actually sequence genomes, next generation sequencing, and then go on to changes that, the types of changes that actually cause variation within genomes. So how are genomes actually sequenced? Well, to start with, the basic problem that we have is that genomes are very large, but the sequencing reactions that we can use at this point in time, produce only short reads. So for instance, the human genome is around 3 billion base pairs long, a little bit longer, but the length of a good Sanger sequencing reaction is actually only about 1,000 base pairs. And putting all these sequences together that we need to basically match the length of a human genome or the genomes of other organisms, which can be equally large or even larger, is very challenging. So, and we can't just get away with producing one single, dividing the 3 billion base pairs into single stretches of 1,000 base pairs, because one, their sequencing produces errors. So we need to actually cover each base in the genome by multiple reads to make sure that we have a good high quality read at that position. Also, we need the way we basically stitch our sequences together is by the sequences overlapping with one another. So we can actually make sure that they're part of the same genome region. And I'll show you guys a little picture of overlapping sequences so that this makes more sense. And this all means that an enormous amount of information has to be managed. So here's a basic outline of genome sequencing. And this is, on the top panel here, is essentially the approach that was used about, now 20 years ago, more than 20 years ago, to sequence the first animal genomes, such as the Drosophila genome and the human genome. And so we start out with the genomic DNA, multiple copies of, thousands of copies, millions of copies of whole nuclear genomes. And then these genomes are cut into fragments. And at that point in time, the standard procedure was to clone them into little vectors. So these are derived from bacteria or from viruses. And they're basically little mini chromosomes that we add our unknown piece of DNA into. And the problem with sequencing unknown DNA is that when we actually don't know what primers to use, right? So remember when we talked about Sanger sequencing, we needed to have a primer that was binding to our sequence of DNA. And by putting them into these vectors, we solve that problem, because we now know that the flanking sequence of our unknown gray genome sequence here is actually known, because we actually know the sequence of these vectors, and we can use that to initiate sequencing reactions from either end of this inserted sequence here. And then once we have those sequences, we put them together into so-called contigs, so pieces of contiguous sequence that are pieced together by virtue of matching overlaps between sequences. So this type of technique has been largely replaced by so-called next generation sequencing. And this is sort of the approach of sequencing that we will go a little bit more into detail here. And the basic principle is that we still start out with the genomic DNA, and it is still being fragmented, but we directly sequence those fragments without actually having to put them into these libraries of vectors, into these mini chromosomes that are derived from bacteria or viruses. And then once the sequences, those individual sequence fragments have been sequenced, the principle is the same. We're again use overlaps between sequences, and in this figure you can actually nicely see the matching sequence between these longer stretch of sequence. The dots just represent sort of a longer stretch of sequence that is not being shown in this cartoon. And again, these overlaps are used to form these contigs. So the other prevalent method that is being used today is so-called shotgun sequencing. And what we mean by that is that the genome is randomly sequenced. We take a genome, we have many genome copies, and we cut those copies into random fragments. And then we make a library of these cloned fragments. So it could either be put into these vectors, or we could sequence these fragments directly with next generation sequencing. And then after the sequencing has been done, we again need to go through these steps of overlapping the individual sequence reads into smaller contigs, and then finally stitching those contigs into larger scaffolds and assemblies. And we're not going to talk about the ins and outs of this latter process here. Shotgun sequencing is in contrast to earlier approaches to sequencing, in which researchers basically used a known starting point on the genome, for instance, the human genome, and then basically systematically walked out, extended their knowledge of sequence from those known starting points, and adding on like specific fragments of DNA one by one. And this was done because sequencing was too expensive. But as sequencing became cheaper and cheaper, it actually turned out that a more efficient approach is actually not to care about where those specific fragments that we're sequencing are coming from, but instead to basically sequence the entire genome and then put those fragments together, right? So it's kind of the equivalent of the Old West philosophy of shoot first, ask questions later. So we're just going to shoot it all up, sequence it, and then we're actually trying to make sense of the pieces afterwards. And so again, building sequence contigs, right? So if we have our reads, in this case, they're very short reads for this example, and we align them, and aligning simply means that we put the identical sequence on top of one another. And if we have enough of these sequence reads, so in this example, it's actually a very extreme case where each sequence is only offset by about 2 base pairs, we can be very confident that these pieces actually all belong together and can be stitched together into a longer stretch of sequence that we call a sequence contig. So one important measure that is used to describe sequencing effort, basically meaning the amount of sequence per length of the genome, is defined as coverage, or is referred to as coverage. And we define coverage as the number of reads times the average read length. So that is our numerator. It's basically the total number of bases that we sequenced, right? Because it's the number of reads times how long each of these reads are on average, gives us the total number of bases that were sequenced. And the genome size, and this is then divided by the genome size to basically normalize the sequence effort by how big the genome is that we're actually trying to sequence. So for instance, if we're taking a human genome, which has around about 3.4 billion base pairs, and we were to sequence 10 million reads with 500 base pairs each of length, this would result in a 1.47 fold coverage of the genome, of the human genome. However, you got to remember that with shotgun sequencing, the actual sequencing is a random process. So we cannot actually predict which specific regions will be sequenced and to what depth. So actually, to just pick up this example here, if we have roughly only 50% more bases sequenced than the genome is long, we are bound to end up with spots in the genome that just by chance were not sequenced at all. Other spots might have just received one sequence, so not enough to actually get a confident read. And then there's going to be a few spots where the sequences are just by random chance piling up high. So in order to actually get a confident read of a genome, we need to up that coverage a lot more. So, standards right now are probably like 20 to 30-fold coverage for a human genome in order to read all sequence variants with high confidence. The other sort of basic distinction that we need to talk about is resequencing versus a de novo assembly of genomes. And that makes a big difference in terms of like costs and ease of how to get like confident genome sequencing. So for instance, in the case of human genomics, we have a very well characterized and assembled human reference genome. And in that case, we can take short reads and we can simply find the spots in the reference genome where those reads actually map to and belong, right? So in that case, it doesn't really matter that we have this little gap here in this cartoonish example where we don't know where the two contexts are actually not linked to one another, because we know where each of these reads go into the genome. We know that this context is to the left and not that far apart away from this larger context here. And once we have a reference genome, this actually is an easy and cheap way to basically score variation in genomes, right? So any sort of personal genomics that is being run on people today for medical or just for basic interest reasons, it uses a form of resequencing and therefore brings down the costs and and ups the reliability of those sequencing efforts. So when we have a de novo assembly, like assembly from scratch, we can use our short reads and basically find the best contigs that we can come up with and form those contigs. But if there are gaps in between contigs, as they're invariably going to be, then we end up with questions such as, which chromosome are those contigs on? What is the order, right? Is this contig here to the left or to the right of this big contig here? Or is it maybe even on a completely different chromosome? So you guys are actually living in an extremely exciting age of biology, and part of that is really driven by the fact that genetics, like genome sequencing or sequencing in general, has become incredibly inexpensive. So this is a graph that's put out every year by the National Human Genome Research Institute. And it basically traces sequencing costs. So this graph here shows the cost per raw meaning that there is no bioinformatic processing done with it, so which also costs money and time, of million cost per raw million base pairs, so megabase of DNA sequence. And so what we have here on the y-axis is actually the cost on a log scale, right? So here is in dollars. So this is 1 cent. Here's 1/10 of a cent. Here's $1. Here's $10 million. Well, it is $10,000. No, 10 million, $10,000. And It sounds about right. So I started my PhD in the year 2000, and it cost quite a bit of money to sequence DNA back then, although the costs had already gone down quite a bit. And then over the next 7 years, sequencing costs actually went down. And remember, this is a log scale, so a linear decline means an exponential decrease in costs. And it actually followed Moore's law, which is a law or a principle that was established in computer science that basically says that the amount of processing power that you can buy for the same amount of money basically doubles every year or every 2 years. I forgot what the actual number is, but it's basically an exponential decline in costs per sequencing or computing power. What happens in 2007 is that different types of sequencing technology are coming online. And we'll talk about this type of sequencing technology. And it leads to an over exponential decrease in sequencing costs with a few jumps in here. And things are now still decreasing, right? Remember, we're looking at a, well, somewhat plateaued with probably future decreases, but the amount by which sequencing has become cheaper is just staggering. So we can also look at this as the sequencing costs per human genome, just expressed, basically taking those numbers from the earlier slide and put them on the, normal, standardizing them by the length of a human genome. So we basically started out with $100 million in 2001, and right now we're around about at $1,000 per genome. And I think, I still have to look this up, but there is actually an offer out there that some company actually offers to give you a 30-fold coverage genome sequence of your genome for around about $300. The only problem is that you're basically selling your soul or your DNA to that company. So what this all means is that, yes, we're producing lots and lots and lots of sequence data. And most of it actually goes underutilized, right? Because there's so much data, we really don't know what to do, or, well, we kind of know what to do with it, but there's so much of it that many, many other questions could be asked with that same data. And what this basically means is that you, as budding biologists, basically need some knowledge in how to deal with large data sets and working with those data sets on computers. And so the biology department, we've been lucky enough to initiate a set of courses that actually helps in the analysis of these large-scale sequence data sets. And so 2 new courses have already been offered and are being offered right now and during the next year as well. So we have sequence analysis, a class that I actually teach. Prereqs are this course or cell and molecular biology, and I will teach this next time in the fall of 2020. And then genome analysis, taught either by Matt Zingraf or Dan Pollard, also in addition requires biometrics, will be offered in the winter and spring of 2021. So something to keep in mind. Okay, so taking a look at the next generation sequencing, the types of techniques or sets of approaches that have really brought down the sequencing costs so dramatically. So again, this is the next generation sequencing workflow, right? Where we have the genomic DNA, we fragment it, and then we've sequenced those fragments directly. So how do we actually achieve this? And the big drop in price is actually due to the fact that this sequencing is happening massively parallel. That means fragments are sequenced in parallel at the same time without handling each fragment individually. So what are some of the basic steps involved in that? So we start out with many genome copies, and we fragment those. And then we isolate those fragments by letting them bind themselves to a surface, right? Because some of the big sort of labor costs and time and money costs come from the fact that before this sort of massively next generation sequencing, each sequencing reaction for each of these pieces had to be isolated in its little own reaction vessel. So either a person or then later robots had to pipette small quantities of genome into individual reaction vessels, and there the reaction, the sequencing reaction would take place. What the massively parallel stuff is doing is that it's actually doing it on one big old slide. It's actually not that big, but it fits a lot of fragments. And also, this is all running automatically at sort of a nano level. The molecules basically arrange themselves or randomly bind to the surface, but with the right concentration, they're spaced on average in the right distance. So, but binding to the surface is very important because it keeps those individual fragments that each have a different sequence in place. And now we can sort of treat them in parallel, but still know what is happening with each of these fragments because they're not moving around in a solution. And so then the other thing is each fragment in itself is too short to be sequenced directly. So we need to make multiple copies of each fragment. But again, we can do this in place by keeping the copies that were derived from the original sequence together and in little clusters that are still distinguishable from other such clusters. And then there's enough DNA there that we can actually sequence each fragment in parallel and in place. So this is the basic principle by how we achieve massively parallel sequencing. So what are some of the common themes of this massively parallel sequencing? So we start out with the genome, and we have multiple copies of that genome that are fragmented. And then these fragments receive universal adapters. I told you a few slides back that the problem with sequencing unknown DNA is that you don't actually know where to start your sequencing reaction, right? You would need some sort of specific primer. But if you don't know what the sequence is, well, you can't make a primer. And also, you would have to make an awful lot of very specific primers to fit all of those different fragments, even if you knew what sequence they were. So the way to solve it is like cloning them into a vector, but instead here we're not using like a bacterial chromosome, but we're using short bits of DNA that are basically glued or ligated to these random fragments, and that now serve as the starting off points for both binding these fragments and amplifying and sequencing them. So the adapters bind to a solid surface, and this is actually done via base pairing, right? There's little anchors consisting out of single-stranded DNA on the surface that can bind our two sequences. And I tried to use these hatched lines and the solid lines to indicate that these are actually two different sequences. And then we amplify these fragments in place. So copies of this fragment stay close to one another, and copies of the other fragment also stay close to one another. And then we can also sequence those fragments in place. So, and this is really the level of understanding that that I'd like you to have for the purposes of this class. The actual sort of techniques and ins and outs and technical specificities differ between platforms and are also at a level of complexity that's beyond the scope of this class. However, the most common type of next generation sequencing as currently used is done by a company called Illumina. And I will post this link as part of the module for Monday's class and you can sort of watch this video and kind of get a better idea of how this bridge amplification works. It's pretty fascinating. But in the meantime, we're basically seeing the advent of so-called third-generation sequencing, and that has the potential to further bring down the costs. So for instance, by techniques like this mini ion sequencer, and also has the potential to solve the problems that we're running into by having very short reads of just about 1,000 base pairs. So platforms such as Biol sequencing actually use single, and I believe actually the nanopore technique as well, use of single molecule sequencing, and thereby achieve sequencing that can actually produce sequences that are tens of thousands of base pairs long. And that, of course, right, if your little genome fragments are now no longer like 500 or 1,000 base pairs long, but 10 or 20,000 base pairs long, that of course makes the assembly of genomes easier and also cheaper. You basically need less sequencing effort for the same amount of genome. So there's a lot of work done in this area, and you're basically going to be seeing more advances in genome sequencing, actually making it easier and easier. So the mini ion, for instance, is basically genome sequencing on a bench top, right? It has a USB port. You could plug it in the side of a computer. So I'd like to take a little break and basically give you a preview of our first discussion paper. And the discussion paper is the first complete genome comparison of cancer cells and normal cells from the same individual. So this paper is now a little bit old, but it sort of was the first time that people used whole genome sequencing to compare the healthy cells, in this case skin cells, with cancerous leukemia cells from the same patient. And it's still a good paper because, being the first study of its kind, it actually lays out the technique and the challenges of the technique. So the first principle, it's in a very accessible manner. Well, somewhat accessible. You be the judge. So why are they doing this comparison of skin cells and leukemia cells? Well, the thing is that most cancers are actually caused by somatic mutations. So, and what we need to distinguish between are the gametes that sort of form the new individual, and then pretty early on in the development of animals, the gonads are squared away, and sort of the germ line, so-called germ line, the cell lines that then produce new gametes to form the next generations, actually put in their own tissues away from the rest of the body, and then the rest of the developing embryo goes on to basically form the soma, the body. And these cells don't actually, contribute to reproduction. So any mutations that happen in these cells are actually never passed on to the offspring of animals. And in this case, right, the soma differentiates, and at one point we're forming cell lines that lead to skin and cell lines that lead to the blood cells. And along the way, mutations will occur, and we call those somatic mutations, right? So just errors that happen during DNA replication. And some errors will be specific to the skin, others will be specific to the blood. And in the case of blood cancer, leukemia, well, some mutations were actually not so benign and actually started causing the cancer. And by comparing the genome of the skin with that of blood, we actually can start narrowing down the types of mutations that are likely causing cancer in this particular patient. And the reason why they choose the tissues from the same patient is that, well, the only mutations that differ between skin and blood are the somatic mutations. We don't have to deal with any sort of inherited variation that might come across if you compare the cancer genome with the human reference genome, right? Because that human reference genome was taken from persons that, yeah, they probably didn't have blood cancer, but they also might have had all kinds of other variants that are not cancer causing, but that are still different from the cells of this patient. So more on that, and I will post the paper. Okay, so one other piece of information is sort of a set of definitions here. And one rule of thumb that holds up in most cases is that individuals of the same species tend to have the same genes in the same positions. So for instance, the gene that causes the ABO blood type sits on chromosome 9 on the arm Q and in the region 3-4 here. So roughly right around here is where we find the ABO blood type gene in the human genome. And of course, we find it on both a homologous chromosome. So we define as locus the location of a gene on a chromosome, and a genetic map is basically an atlas of positions of genes in a genome. So another really important statistic to consider when we talk about genomes is actually this question, right? What fraction of the genome actually consists of genes, and what fraction consists of other DNA? All genes are composed of DNA, but not all DNA actually constitutes genes as we most commonly define them. And also the percentage of the genome that codes for protein coding genes actually varies a lot between organisms. And the general trend is that so-called more complex organisms contain more protein coding genes, but they actually make up an overall lower percentage of the DNA. So here, we can basically approach this also with the following question, right? So if we have a piece of genomic DNA, and there is a G in this genomic DNA, and we can ask, right, does this G actually have a function? And we can ask a few questions of whether that's true. So is it actually transcribed? Is it translated? So it would be part of coding sequence. Is it part of a recognition site for transcription factors? So does it play a role in gene regulation? So when it comes to, when we ask those three questions, for bacteria, most of the genome actually has a function, right? So in organisms that are well studied, like E. coli bacteria, that is indeed the case. If it comes to organisms with smaller genomes, such as roundworms or Drosophila fruit flies, the proportion of the genome that is actually exonic is actually just 1/3, but about 65% of the genome is actually transcribed. So it may actually serve a function. Might not be as coding sequence, but it could also be as functional RNAs. And in humans, it's actually pretty similar when it comes to the amount of genome that is being transcribed and may be functional in some way or another. But only 2% of our genome is actually exonic. Right? So that is this small proportion of the entire genome. And 70 up to 70% of our genome may actually be is actually transcribed and may have some sort of function. But the big question mark is, well, is this true? And what are these functions actually, and how is this all working out? Also, we can basically turn the same type of information and ask a different questions. So if you take any base in the genome and at random and change it, is there a functional consequence, right? So in our case, we take our same exact G and we asked, well, if we change this G, does this affect the phenotype? So, and the answer will again differ by the types of organism, right? So if most of the genome is exon and bacteria, there's a good chance that it may actually cause a change in phenotype. But however, there's a few caveats here, right? So it could be a third base change, so a change that does not result in, so basically a silent mutation that does not result in change of amino acid. It could be a conservative substitution, meaning that it is actually a change to another amino acid at that codon position that is very similar chemically and may fulfill the chemical role of the original amino acid in a very similar way, and therefore not really cause a sort of phenotypic change. And there might be redundant information, right? So there could be a second gene that does almost exactly what this mutated gene did, and therefore we're actually not seeing any phenotypic changes. All those things also apply to other organisms, right? So we could, you know, have a change, but it could also be, again, a synonymous substitution. It could be a conservative substitution. And when it comes to redundant information, well, there's other things in these organisms. So for instance, multi-gene families that have genes of similar functions, we have duplicated pathways. And now that we're dealing with diploid genomes, right, with pairs of homologous chromosomes, the mutation could actually be a recessive allele, meaning that on the other chromosome, there's still a functioning allele that can sort of jump in and take over the function of the allele that suffered the mutation. And so we refer to these things as various forms of redundancy and robustness. So how do genetic changes and result in morphological differences? So we can talk about a few types of different changes. So if we start out with changes that are very localized, so small scale changes, there's different classes of these. So if this is our normal or wild type sequence, and that is of the comparison against which we compare the effect of a mutation. So we could have a base substitution. That means the T is exchanged for a C. Right now, the sequence reads TCCAAT instead of TCTAAT. A base could be deleted, right? So in this case, this is symbolized in this alignment by a dash, but in the actual chromosome, right, we would simply miss this base here, and it would simply read TCAAT instead of TCTAAT. Or we could have a base insertion. So we basically added an additional base and made the chromosome one base pair longer. And so we're looking at TCTAAA triple AT for the same reference sequence. Then we also have sort of largest type of insertions, so-called copy number variation, in which we have larger bits of DNA that are being duplicated. And of course, deletions could also involve larger bits of DNA. So if we look at point mutations in coding sequence, we can distinguish between different types of mutations with different consequences. So here is amino acid sequence that is encoded by this wild type sequence. So threonine, lysine, arginine, glycine. And now we can look at different types of changes. Again, the first one is it could be a synonymous mutation, right, or a silent mutation. What that means is that in our original sequence, we had AGA coding for arginine. And in this sequence, it's now AGC, but it's at the third position. And there's redundancy here. And now we still have an arginine being encoded by a sequence called AGC. Then we have mutations that we refer to as missense mutations. And in that case, so here we have a second position that changes. We changed the arginine to a lysine. But we would refer to this one as most likely a conservative missense mutation. Conservative meaning that the amino acid residue that's being exchanged here, right? One is exchanged for another one that is chemically pretty similar and is likely to fulfill the same function in the protein. However, that's just a rule of thumb, right? So chemical similarity is not necessarily a guarantee that there's no drastic phenotypic effect. It all depends on the specific position in the protein. And then non-conservative missense mutations basically exchange one amino acid for another one that is chemically dissimilar. And again, the same thing applies, right? There's no guarantee that this non-conservative change will actually result in a drastically altered function, a loss of function of the protein. It all, again, depends on where this is situated. If this is in the active center of an enzyme, most likely this is going to cause wreak havoc. But if it's on the outside of the protein and does not interfere with protein folding or interactions with other proteins, it might actually be not of much phenotypic effect. And then the third type is nonsense mutations, and that is basically the addition of a premature stop codon that then results in the stop of translation. And here's just another way to look at this and to add some other types of mutations. So for instance, here is a gene. The exons are shown in dark green and the introns in light green. And the sort of pinkish regions are actually regions of exons that are involved in the active site of this enzyme here. And the mutations that we're seeing up here in this alignment are also mapped. Some of them are mapped to the actual protein. And what we have here is sort of a classification of these different types of mutations. So we could have a null mutation, meaning that basically leads to a loss of function of the enzyme. A silent mutation will have no phenotypic effects. And a leaky mutation may actually lead to the impairment, but not of the function of the enzyme, but not to the complete cessation of function on this enzyme. So here's mutations in regulatory regions can actually be very influential. So this one here may actually cause for this gene to no longer be transcribed at all, and therefore resulting in, basically in the loss of this particular gene copy, resulting in a null mutation. These two mutations here in this cartoon are located smack in the active site of the enzyme and would probably alter its function in a way that's no longer functioning. This M4 here, so if it's described as leaky, is kind of at the boundary of the active site to the rest of the protein, and it may interfere with protein function somewhat, but not so drastically. But again, this is just a cartoonish version, right? Some of these mutations that are sort of in the boundary region may very well have very bad consequences, or may not have any consequence at all. And then the M5 is referred to as a silent mutation, right? We may see an exchange of an amino acid, but it doesn't actually change the protein function. And then other types of mutations. So here's one in the intron, right? One might think that those types of mutations are actually not of any consequence because the intron is not being translated. However, this mutation is located of one of the intron boundaries. And what it's supposed to symbolize in this cartoon is a case where mutation affects the ability of the cell to splice out the intron. And introns that are not spliced out are, of course, extremely harmful, right? So if this is being translated with intron, we're going to have frame shifts, we're going to have amino acids being translated that are not supposed to be there, and we're probably going to have a premature stop codon somewhere in here. So all kinds of bad stuff will happen if you try to translate introns. So again, silent mutations or synonymous mutations are basically ones that don't result in an amino acid change. And we have different forms of degeneracy, right? Most of it at the third position. So for instance, here for proline, at the third positions, both U, C, A, and G all code still for proline. In some cases, we have multiple options for the first position, but in no case do we have multiple options for the second position. And then this is the idea of a conservative versus non-conservative amino acid substitutions. And what you're seeing here is basically a graphical representation of a matrix of probabilities that was put together by Margaret Dayhoff, one of the pioneers of bioinformatics. And what she did, she and her colleagues did, was to basically compare known sequences of proteins and look at how likely it was that protein sequences would change between different organisms. And the important thing here is they did not look at mutant or sort of harmful mutations in proteins, but they looked basically at accepted mutations or mutations that were accepted by evolution. Right? So if like a mouse and a human differ in their protein sequence at a certain gene, and both sequences show the wild type sequence for mice and humans, then any sort of those differences must obviously be working. And so we call them evolutionarily accepted mutations. But what she found was that well, it's probably something that's not too surprising. So the distance in this graph here basically is negatively correlated to the probability of an exchange. So for instance, glycine and tyrosine are much less likely to exchange for one another than aspartic acid and glutamic acid. And this correlates very well with sort of two chemical characteristics, the size and the polarity of the residue. So small polar amino acids are much more likely to be exchanged for one another than small non-polar ones for a large polar one. Then also the other type of variation that can result, a genetic variation, like sequence variation that can result in phenotypic variation, is variation in regulatory sequences. So here's an example from humans. And in certain populations, the difference between somebody having brown versus blonde hair is actually caused by by a nucleotide substitution in a regulatory region, in the cis regulatory module, about 350,000 base pairs upstream of a gene called KIT ligand. And this cis regulatory sequence results in higher level of expression for KIT ligand, and this sequence results in lower expressions of KIT ligand. And KIT ligand is part of a whole system that basically influences the deposition of pheomelanin, light pheomelanin versus dark eumelanin in the hairs and in the hairs of mammals. And we already talked with regards to the squirrels about this system here, agouti, MC1R, and then further downstream molecules that then mechanisms and proteins that result in either expression of fear or eumelanin. KIT ligand is sort of an additional pathway that also impacts the same mechanism downstream. And so if KIT ligand is overexpressed, is expressed to a higher level, then we're basically seeing more eumelanin being deposited. And if KIT ligand is expressed at a lower level, we are seeing more pheomelanin being deposited. And again, this is not a change in KIT ligand itself, but just in the regulatory regions that lie upstream of KIT ligand. So this, what we just talked about, was a very subtle change in the genome. Now we're going to talk about a very drastic change in the genome, and that is polyploidization. So this is something often observed in plants, in that through errors during mitosis or meiosis, during mitosis mainly, we're seeing we're basically seeing cells not dividing after replication, and now we're ending up with double genome copies in those cells. So if we go from a diploid organism to after a failed mitotic division, we may end up with a cell line that is now tetraploid, and in plants that could result in in a novel clone, or being turned into a seed or pollen, and then being propagated. And here's another case, hexaploid. So polyploidization simply takes the entire genome and makes sort of additional copies of the entire genome. And that, of course, is a massive scale change. At a smaller level, duplications can also increase the DNA content and increase the gene content, and with that also sort of additional functions of those genes. So a really famous example for these multi-gene families is myoglobin, are the hemoglobins. And hemoglobins are related to myoglobin, right? The globin in muscles. And myoglobin and hemoglobin shared a common ancestor many million years ago, sometime in the dawn of the vertebrates. And starting with vertebrates with jaws, so basically all kinds of fish that are not lampreys or slime eels, we're seeing a duplication. Well, we see myoglobin splitting into the alpha and beta globin precursor, and then we have a split of the alpha and beta globin into alpha precursor, into alpha and beta globin, that is now found in all vertebrates with jaws. And then we, in humans and in other groups, we saw further differentiation of both alpha and beta globin. So for instance, some of these genes are actually fetal alpha and beta hemoglobins that are expressed in fetuses and then no longer expressed in babies and older organisms that see the expression of sort of the standard alpha and beta hemoglobin. All the while myoglobin is, of course, still around and being needed and expressed in the bodies. And then I'd like to end this lecture with a really cool example of how gene duplication results in new functions. And this is color vision in primates. So this is actually not somewhat easy and not complicated, in other senses quite complicated. So old world monkeys, which includes us and this chimp here, are trichromatic. So we basically have receptors in our eyes that can detect wavelength, receptor S, for instance, here in the blue light, but then we also have receptor M and L. So M has its peak reception in the green spectrum, and L somewhere in the orangish red spectrum. And with these three colors, just like on your TV, old school TV, you end up with blue, green, and red, and the ability to basically make all the colors that we can perceive. New World monkeys are dichromatic. They basically only have the ability, well, the base state is the ability to only see in the blue color spectrum, and then in a color spectrum that is somewhere in the red or yellow, red, yellow, green, but just a single peak. It's more complicated, and we'll get to that. So the reason for this difference is that there was a duplication of the ancestral sort of red-green receptor in the lineage that led to Old World monkeys. And Old World monkeys now have both an L and an M copy, right? The L copy seeing in the orange yellowish, and the M copy having its highest sensitivity in the green spectrum. Both receptors, L and M, but not S, are on the X chromosome, meaning that in genotypes that are XX, right, so let's call them females, but things are a bit more tricky and difficult, and we'll talk about that, and XY have 2 copies each, whereas XY genotypes have also, let's call them males for now, even though it's more complicated, on the X chromosome, and no copies on the Y chromosome. And that is actually one of the reasons why people with XY genotypes have a higher probability of color blindness, because if there is no redundancy, if a recessive mutation occurs on their only X chromosome. New World monkeys have only one single receptor. However, in some female New World monkeys, so XX with XX genotypes, we actually see instances of trichromatic vision. And the reason for that is that there are sometimes different alleles for this receptor, and those different alleles may have color reception in different wavelengths. So there are some females, if they're homozygous, they are dichromatic, and males are always dichromatic, but they might be dichromatic for blue and some allele, whatever they got, and those alleles may actually have different peak, wavelength reception at different levels. However, only females, right, since males only have one X chromosome, only females can end up in a situation where they're actually heterozygous, have 2 different copies. And in that case, we can actually have trichromatic vision in New World monkeys. So this is a really cool, fascinating system. Okay, thank you, everybody. We'll see you on Monday.